Yes.
I was wondering about comparisons, pairwise comparisons of just pairwise correlations, test scores.
That was already more commitment than I would have ever been able to contemplate. I was just like trying to write down their names, and I already like gave up so much. I can't even bother. I wouldn't have imagined like having a pairwise correlation already like blew my mind.
It wasn't that many students.
I don't know if you heard that random motorcycle. I live in London, so it's like there's always some background noise. I hope it's not too much. It's peak rush hour at 5:30, so I hope I could keep it to the minimum.
So I think it's 12:32 right now at 9:32 Pacific time. Jennifer, should I go ahead or Noah? Do you want? Should I just start the introductions?
Yeah, you can go ahead, unless Noah wanted to say anything. I don't think so.
Okay.
Yeah, just thanks everybody for being here, and thank you, Katia, for organizing this event.
Yeah. Big thanks to Mahan and Alison and Chadi all for agreeing to speak here. This SMTPB mini-symposium focuses on theory in understanding viral dynamics and evolution. And so this is, of course, kind of the Society for Modeling and Theory in Population Biology. And over the last decade, I would argue that there's been just a ton of modeling done, especially during the SARS-CoV-2 pandemic. So a lot of infectious disease modeling, but I would argue that there's quite a bit less of kind of theoretical developments in the field, per se. So for this mini symposium, I've invited 3 speakers who I think really have been doing groundbreaking work in terms of actually developing theory for kind of broad understanding of viral dynamics and evolution, and with really kind of broadly applicable kind of broadly applicable to different viral systems, even if their work was really kind of focused on developing a theory for data that's available for a specific viral system. The bigger kind of principles and theoretical understanding gained from that analysis, I think, is more broadly applicable. So the 3 speakers today are Mahan Ghafari from University of Oxford, Alison from University of Washington, and Chadi Saad-Roy, currently at UC Berkeley, but soon to be at University of British Columbia. And the work of these 3 speakers that they'll be talking about today, respectively, will get at longer-term viral evolutionary patterns and how we can understand them, the relationship between viral genotypes and phenotypes, especially in the context of cellular co-infection, and viral adaptation and how it affects the host population. So topics that, again, have very broad applicability across different viral systems. And with that, each speaker is going to give about a 15-minute talk, and then we'll have a few minutes for questions, and then we'll move on to the next speaker. So Mahan is going to be the first speaker, and then Alison, and then Chadi. And then we'll just open it up to kind of a discussion for 10 or 15 minutes, focused on just theory in the field of kind of infectious disease ecology and evolution with specific viruses. Okay, go ahead, Mahan.
Let me know if you could see this right screen, hopefully. Can you let me know, please? I can't see you. Sorry, I have one screen.
Yeah, yes, we can see it.
All right, good. Thank you, Katia. It's really great to be virtually here. And yes, like Katia said, I'm Mahan. I'm a Wellcome Trust Early Career Research Fellow at Oxford, and I'll be looking at, for the next 5 years at least, developing a method for systematically estimating evolutionary rates and reconstructing viral histories. So let's just start with building up the the sort of momentum and understanding why estimating evolutionary rates matter. Suppose you have an outbreak with an unknown pathogen, cases grow rapidly. Let's actually make it an interesting outbreak. Let's say it's a novel coronavirus, and you collect sequences, swab samples of infected individuals, and you collect the viral sequence. And by comparing the genome of those samples with respect to the earliest sample, you notice a clear pattern in the data. Well, it usually doesn't look that good, but viruses evolve very rapidly, so you could get this sort of a clock signal in the data after a couple of months worth of evolution. And the reason why it's helpful is because you could then make ancestries, like make phylogenies of the virus, and that could then tell you something about the movement of the virus. And actually, say if you happen to have collected, say, I don't know, a couple of samples that are only a few days apart, but you look at the virus and you already see a few dozen mutations. I hear an echo of myself. I don't know if it's someone on not mute or something. I don't know. I hope you can hear me okay.
Yeah, if everyone could please just check whether they're muted. I don't think I have the ability to mute anyone.
That's okay. Anyways, I hope you can hear me. But yeah, that could tell you something about the viral movement. And if you have a set of samples early on and they are already somewhat divergent relative to one another, and you could say, oh, I know beta coronaviruses, evolve at this certain rate, you could then get a better sense of, well, how long the virus might have been circulating undetected. And that could then give you some better sense of how long it may have been circulating undetected and what's the true prevalence is. Well, you could push it one step further and ask, well, what's the closest common ancestor of these viruses with respect to their natural host? And again, with the clock, if it were zoonotic origin, let's not talk about that in this context, but if it were, you could then, from this notion of clock, you could say something about the timing of the spillover. But the challenge with these clocks is that they're tricky to measure. And I think my favorite example is with the Ebola outbreak in Sierra Leone back in 2014 to 15, where early estimates were like 2 to 3 times, reporting rates that were 2 to 3 times faster than later in the outbreak. And that led to conflicting statements at the time where some people were saying, well, Ebola is evolving very rapidly, our vaccines are going to become ineffective. Then later studies said, don't panic, it's not evolving very rapidly. And then later studies, which sort of gathered all the available data, genomic data across multiple waves of infection in Sierra Leone and elsewhere, they said, well, no, it's actually all the previous ones were wrong. And these differences are just artifact of how rates were measured. And fast forward to 2020, early on in the SARS-CoV-2 pandemic, we heard similar statements from various people saying, well, we shouldn't worry when a virus mutate. These mutations rarely impact outbreaks dramatically, which in fairness was largely true until we saw these variants of concern emerging, which were highly mutated, alpha, beta, gamma, at the time in mid-2020, early 21, that really shifted our perspective of what's the mode and tempo at which viruses evolve over time. And I think that was the underlying thesis of my fellowship, was to really trying to see if we can develop a unifying framework for understanding how rates vary and what are the implications of that for reconstructing evolutionary histories. And I want to divide up my talk into 2 components. So I'm going to talk a little bit about earlier on the biological factors that could lead to rate variation, and then later on the methodological factors. Back to SARS-CoV-2 again, this is an animation, and I'm going to present in a second, from NextTrain, where the black line is representing the clock rate of SARS-CoV-2 in the first 4 months of its emergence, and the rate is something like 27.8 substitutions per site per year. And as you see in the first rate, there's a sudden drop down to 20.2, and then right after the emergence of Omicron, you see this sort of sudden burst and increase in inferred rate. So at least the take-home message is using a fixed rate is going to be flawed. The rate can vary in very peculiar ways, and if you just use the fixed rate, which, believe it or not, 89%, 90% of the literature is doing that, is going to lead to flawed inferences of evolutionary timeline of viruses. And what we and others have shown over the last few years is that the real picture is quite a bit more complex than that fixed rate. And in fact, what we saw was that within a given lineage, say alpha shown here in purple, you get that linear accumulation of mutations, but then these lineages are separated by these saltation, these jumps in evolutionary distance. So going from, say, alpha to Omicron BA1, there's sudden bursts of extra mutation that BA1 has relative to alpha, which cannot be explained through this normal gradual picture of mutation accumulation. And sort of to understand this whole process of what causes rate variation, not just saltation, I think, I was looking at this like many others, perhaps in 2020, I was playing with SARS-CoV-2 genomes. There was so much of it coming through, and I was trying to measure its evolution rate. And I was noticing as I was gathering more and more sequences, looking at more and more sequences, I was inferring a slightly lower and lower rates. And then I was digging up more of the literature. I was looking at papers on pandemic H1N1. Klaus Wilkie and others had papers back then on PH1N1 also reporting rate decline. And to me, that was like an early indication. Well, because I was seeing even within my data that the tip branches of the tree tended to accumulated more mutation relative to the deeper nodes, that was an indication that probably purifying selection is at play, right? I mean, my favorite paper at the time was like the Nicolaisen Design 2012 paper, but then Katia and David Rasmussen had a lovely paper recently, but really looking at investigating the role of purifying selection, not just on impact on its sort of evolution rate, but also other phylogenetic parameters, the tree topology, and so on. But anyways, this was sort of, to me, like purifying selection could give rise to a pattern of an exponential rate decline where the exponent of the decline could be determined by the strength of that selective coefficient S, the deleteriousness of the mutations. But I think the picture is more nuanced. I think there's a lot of caveats when you want to, go from that picture to then explaining what we see. And I'm not going to be able to talk much about that. Just a signpost that, there's caveats, and Javier, one of my students, is looking at that at the moment. But perhaps the more interesting bit, or at least the one that a lot of people were looking at, was these saltations, like I said earlier, and it was earliest documented example was the alpha. And long story short, again, I'm not going to talk about the details. We sort of reviewed all the available evidence back in 23, where all the evidence really pointed to chronic infections, and that it was almost like a split image in terms of the types of mutations that the virus was acquiring during these chronic infections, and where the virus was acquiring them was very similar to the sort of things we saw with the emergence of variants. And with Daniel Wiseman and others back in the time, we looked at trying to understand what sort of fitness landscape can best explain this sort of saltations we saw with the emergence of the first three variants, alpha, beta, and gamma. And we concluded that sort of complex adaptation, so this fitness landscape in pink on the bottom right, whereby a chronically infected individual needs to acquire a couple of mutations that are each individually neutral deleterious. But if that individual happened to have collected, say, I don't know, K of them, then together those K mutation confer a fitness advantage. And that turned out to best explain the available data at the time we had in terms of the timing of the emergence of alpha, beta, and gamma. That was a purely theoretical exercise, if you like to call it that way. I'm going to come back to this a bit later, but that really motivated pretty much the last 3 years of my life, where I was just trying to sort of characterize the dynamics of these chronic infections. Well, first of all, beginning with understanding how prevalent they are in the general population, and identifying that, about 1 in 1,000 or so of all COVID infection can persist for longer than 2 months. Well, knowing COVID is generally an acute infection, 2 months or longer is quite a substantial period. And then actually, it turns out most of these infections were in the elderly population and in males. And because we were looking at a household transmission study, it was in fact the largest surveillance study of its kind, we could establish whether any of these chronic infections led to onward transmission. And within the 100 or so pairs that we had, 100 or so chronic infections over 2 months we did not find evidence of onward transmission. So probably the good news is that the transmission is likely rare. But curiously enough, within those persistent infection, there was a wide variation in the estimated evolutionary rate of the virus. In fact, not all of them had accelerated rates. And in fact, maybe up to about 5% or so of them really had evidence of rates being so high to the same level of like that could give rise to an Omicron-like event. I don't know if Daniel is in the audience. He would always come up with something about my arguments being flawed, but this is my first time actually putting these numbers together and making a statement about that theoretical exercise we had back in 2022, because there in our modeling framework, we had this quantity called the PF, which is the probability of a chronic infection leading to an emergence of a highly divergent variant being less than 1 in 10 million infection event. And I think the evidence we have from these studies that I just presented over the last few years seems to corroborate that, seems to support it. And I think this is where I like theory a lot and why I love to do theoretical work, and I like SMTBB communities because we want to make sense of observation. And yeah, anyways, enough said. I'm going to now switch gears to talking about something completely different. So talking about methodological factors that could influence evolutionary rates. I know for the phylogeneticists in the audience, modeling rate heterogeneity among sites in a genome of an organism using a gamma function is quite well established. You probably all know about it, developed by Zhiheng Yang back in late 1990s. It turns out it was so popular, everyone wanted to use it, and then they use a simplified parameterized version of a discretized gamma, which would, approximate that continuous gamma function. But that, and importantly, by discretizing it into 4 rate categories, you would attribute equal probability of sites evolving under each of those 4 different rate categories, basically trying to capture rate variation across the genome. So not every site evolves at the same rate. It turns out, as we showed recently, this work was really led by Luca Ferretti, Matthew Hall, and others, that this discretization with equal probability of gamma leads to an interesting, curious, purely methodological artifact, really, I think it's fair to say, of branch lengths of these samples being inflated as you add more sequences, and that's sheered simply by the fact that you just discretize the gamma, and free rate being an alternative model which doesn't have this equal probability assumption per rate group doesn't suffer from this branch expansion problem. I was interested particularly in this phenomena for clock rate estimation purposes, and through some simulation work recently with Matthew, I was able to show that again under constant coalescent population size, yes, this sort of discretized gamma, discretization of gamma basically leads to inflated branch length. So pretty much terrible news for the field because we've been using it since 1990s or so. A lot of rate estimations could be flawed. So it's warning, I guess. Again, switching gears to yet another issue, perhaps related to methodology, this sort of scaling of inferred or apparent evolutionary rates, as I would like to call it, with respect to the time frame of measurement. This was really the work that was done by Aris Katsorakis and colleagues back in 2016 or so, where they were like cataloging all these different rate estimates of different viruses, retroviruses, coronaviruses, all sorts of viruses from all the different Baltimore groups you could imagine. And they were seeing this consistent pattern of the rates seemingly declining as the time frame of measurement increasing with a power law decay of -2/3. I was at the time a PhD student trying to figure out the simplest possible explanation of this phenomenon, and the one that came to my mind was site saturation, right? So on the left, think of the simplest possible model of a Jukes Cantor where, ACTG mutation rates are all the same, and you start with an initial homogeneous population, and on average, over a time scale inversely proportional to your fixed mutation rate, you would expect every site to have had, on average, 3/4 mutational differences away from whatever your initial wild type is. And beyond that point of saturation, if you want to measure amount of evolution that have happened in the virus, you're not going to get any further evolutionary change. You wouldn't be able to measure further divergence relative to your root. So that could then, as you can see on the right, give rise to a power law scaling with a slope -1. But then, okay, that was a very boring, simple scenario of 1 rate category. But if you make it slightly more interesting, so would be purple here, shown here, but make it slightly more interesting, suppose there's 2 rate categories, one very rapidly evolving, 10 to the -3 substitutions per site per year, one very slow evolving. And those 2 proportions are, distributed in such a way that the mean rate is the same, compared to a single rate category. What ends up happening is that once you hit the saturation limit for the rapidly evolving sites, you get this blue scaling you see on the right-hand side, where it's not quite power law, but the rate sort of declines down all the way to that slow evolutionary rate over longer time scales. And that's really fundamentally the logic behind this prisoner of war model, which I developed back in 2021, where the idea is simply that if you have now a number of rate groups going all the way up from the very rapidly evolving sites, mu max, down to the very slow evolving groups of sites, mu min, and they are somehow distributed. I just picked exponential because it had one parameter. It was the easiest. Through chronological saturation of these sites at different rate groups, or starting from the fastest one down to the slowest one, which never saturate, you get this sort of scaling pattern shown with the red line, which mimics the empirical observation that was made back in 16 by Isakun and Katsurakis. Anyways, I'm going to wrap up now. Just a shameless plug here. Sorry, I'm recruiting postdocs. So if any of this stuff that I was just telling you sounded interesting, please get in touch. And with that, I'd like to thank you all for listening. I look forward to your questions. Sorry, I don't know how much I ran over time. I hope not a whole lot.
No, you stayed very much on time. Thank you. Does anyone have any questions?
So I have a comment. Sorry, I didn't. So I just have a comment that this issue of saturation gets rediscovered over and over and over again. And as a historical comment, I think the first version of it is Gingerich et al. In 1983 in the context of paleontology and trait evolution. If I have a chat, I'll put that in the chat. It's just, I think, a case where the same issue comes up on field after field after field, and I certainly felt a little silly when I found out the paleontologists were, way ahead of us.
Yes, it has been discovered. I didn't claim that I discovered it, but I do agree that it has been discussed under the umbrella of scaling. I think paleontologists would have framed it slightly differently under trying to remember the name. I think lineage diversification of things. But yeah, it's been reviewed recently as well. I think I just posted a relevant paper you might want to look at, but I agree.
So I have a little bit of a broader question. So you talked a lot about kind of, your work focused on the SARS-CoV-2, during the SARS-CoV-2 pandemic and all the sequences that are made available there. And so, I guess my perspective is kind of that, there were these questions very early on, the first, 2-3 months of the pandemic about, what is going to happen, right? In terms of, is there going to be antigenic evolution? Can people get reinfected? Is this going to become, more virulent virus? What's going to happen, right? And I think in terms of at least kind of the evolutionary predictions, or suggestions, it was, they were not very good, right?
I'm sure there's one person that's going to say, I told you so. We're going to see these big jumps. I'm sure that, but yeah, I agree.
So I guess in terms of specifically kind of viral evolution rather than kind of viral population dynamics, but viral evolutionary dynamics and adaptation, I mean, there's been some work. I saw Troy Day was on here, on this call as well, so some of the work he's done with Sally about kind of, well, what phenotypes might you expect to evolve and so forth. So, where do you think theory needs to go in terms of actually, before the next pandemic, whatever that virus might be, in terms of actually really being able to be kind of applied kind of on day one, in terms of saying, okay, well, these are the things that we might expect, and what is the timeline at which we kind of expect these phenotypic changes to be occurring?
That's a great question. I don't know how much I could live up to answering it, but I do because I think SARS-CoV-2 was an interesting case because it was really, I mean, it's the only organism for which we have 17 million sequences, right? So we haven't ever looked at anything at such a scale. So I don't know. I think we could revisit some of the old assumptions we have of how viruses should generally evolve. I mean, I like to now think, does flu evolve the same way as, SARS-CoV-2 does? We do know that there's cases of chronic flu, and they also lead to emergence of novel variants. So I think, yeah, I mean, you recently had a preprint that I really like. It was like looking at within host evolution and different scenarios of, I think, House of Cards type models of. So that was really fascinating. I was like, please have more of this because we need some ideas about what are the sort of regimes of what the landscapes sort of, or what sort of adaptations could give rise to these episodic accelerated evolution or decelerated evolution. I think that's where I think the field is exciting. I think that's exactly where I think we need new ideas, like linking PopGen to phylogenetics. I think that's broadly how I see it. And you've been leading a lot of that work. So thank you. But yeah, no, I agree.
Yeah, all these nice things, but thank you, Mahan. Any other questions? Okay, something in the chat from Noah, also about diversification models and paleontology and solid dynamics. Okay, great. Special issue plug. Okay, great. All right, well, thank you, Mahan, and you'll come back at the end for the general discussion. All right, Alison, you're up.
All right, thank you. We're going to be narrowing the scope and the scale a little bit here. Let me bring up some slides. All right, everyone can see this okay? Great. Okay, I'm not going to kind of be giving you a totally wide-ranging description of a lot of research like Mahan just did, but I do want to tell you about an idea that I've been excited about lately in the context of intrahost evolution. So my lab works primarily, about the evolution that happens after a virus gets inside a body and starts replicating. And I've really started thinking about things somewhat differently in the past few years, and I'd like to share sort of what we've been up to. So this has to do with the evolution of drug resistance, specifically in the context of the poliovirus capsid inhibitor called picapavir, which I'll tell you about in a second. But I want to just start with this much more general comment about how resistance evolves that everyone is going to have seen this picture and be really familiar with it, which is that imagine you have a population of viruses and there's some heterogeneity in the population wherein some viruses are susceptible to a drug and others are resistant. If you treat with a drug, those viruses that are susceptible are going to be bound by the drugs. The ones that are going to survive are the resistant ones, and they're going to grow back and cause a resurgent infection. So we've all seen this picture a million times, but I want to highlight something that we take for granted perhaps when we look at this picture, which is that there's this strong association between a viral's genotype and its phenotype. So the fact that this virus's genotype encodes resistant a capsid means that when this drug selects for this resistant phenotype, it also selects for this resistant genotype, and that's how this expands. Now, I think the field has been reaching this. It's not really a new understanding, but a new understanding that is really impacting how we're viewing viral evolution over the, maybe the past decade, wherein we think about viruses intrahost as having frequent co-infection in which multiple different viral genomes can enter single cells, and their genotypes are able to combine and interact with each other in interesting ways. And I'm going to be telling you about this in poliovirus, but this is, happens in influenza and a wide variety of other viruses. So let's think about what this should do to the emergence of something like drug resistance. So if you have multiple genotypes that are present in a cell, they might all be producing some phenotypic component, like imagine the capsid subunit that can ultimately form together to become a capsid itself. If there are phenotypically distinct versions of this capsid subunit inside of a cell, they can form these chimeras of resistant and sensitive subunits that can then, load the genomes that are present in the cell and be exported. And I think that this is really interesting because it's no longer the case that there's strong genotype-phenotype association. What's really going on is that a phenotype is not just your genotype, but also the genotypes of all of your co-infectors. And I think that this has a couple of really interesting implications for the evolution of resistance. The first one being that, if resistance is rare in a population, you may reach a state where, despite the fact that resistant genotypes are present and are expressing their phenotype, they might not actually be expressing it strongly enough in order to sort of have selection be able to see it. So in this example here, you could imagine that a bunch of susceptible subunits that are comprising each of these capsid subunits might be able to be bound by this drug and neutralized despite the presence of resistant phenotypic components. The second thing that I think is important here is in recognizing that even if you do stochastically produce something that is more phenotypically resistant, it isn't necessarily paired with a genotype that matches. And so this has implications in terms of thinking about how these traits are going to be transmitted in future generations. You're not necessarily going to get that major expansion. So this field that is considering how we can use viral social traits in order to target viruses and maybe design therapeutic strategies based around these ideas, I think is really, really exciting. So luckily for us, there are existing therapies that actually work exactly like what I've just described. So one of them is this capsid inhibitor called pacapavir. So it's a treatment for poliovirus. Folks may not know, but we actually have no FDA-approved retrovirals that can treat poliovirus. So pacapavir is really exciting because it actually seems to have some real promise. So this is just sort of a standard picture that shows, if you treat with pacapavir, you get a huge knockdown of the viral yield in cell culture. However, pacapavir, like a lot of different drugs, is vulnerable to resistance evolution. You can get, single mutations in these capsid subunits that confer strong degrees of resistance, such that, if you treat with this drug, it doesn't actually change very much about the viral yield. So this is just a standard antiviral. Why do we think that it is having this susceptible interference property? Well, Carla Kierkegaard's lab, led by Elizabeth Tanner in about a decade ago, did these really exciting experiments in which they mixed viruses that had, resistant viruses. They put resistant viruses into cell culture and then mixed them with varying degrees of susceptible viruses, sort of from no susceptible viruses at all to lots of susceptible viruses. And what they found when they did this is that increasing the total amount of susceptible viruses in a cell actually knocks down the yield of these resistant ones. In other words, what we're seeing is that the susceptible subunits are basically poisoning the well. They're meaning that the resistant viruses aren't able to express their phenotype effectively. And I'll note that the viral yield is going down despite the fact that the total MOI, the number of viruses infecting each cell, is increasing over the course of this experiment. And this is something that you don't see if you treat without pacapavir. The more viruses you add, sort of the more viruses you get back. Okay, so this is really exciting. It suggests that we're able to knock down the viral yield by even in the presence of resistance because of this property of the phenotypic mixing. Unfortunately, I said, there's no FDA-approved antiviral for poliovirus. This sort of had not completely persuasive results in clinical trials. So they did this clinical trial in which they treated people with the oral polio vaccine and either a placebo or pacapavir, and they asked, when did people clear their infections? With the hope being that you might be able to give someone pacapavir simultaneously, and what you should get out is maybe an infection that resolves faster and has less risk of shedding and other complications. And what they found when they did this is that the clearance times between the placebo and the pacapavir group were not all that different from each other. There was this enrichment of very early clears in the pacapavir group, which sounds pretty promising because it suggests that, maybe some people really are benefiting from this drug. But the results became a lot more sobering when they did sort of genotypic tests to try to understand whether resistance was present or not. And they found that among these people who were treated with pacapavir, there was this huge enrichment of resistance among the late clearers. So what this suggests is, despite the fact that we think that these strategies should be able to keep resistance evolution in check because of the susceptible interference phenomenon, it doesn't actually seem to work in practice. So one of the things that we've been doing is trying to build some models that can try to explain these observations. So what I want to quickly talk you through is first, how does, the model that I'm gonna get at to try to answer this question: how does poliovirus evolve resistance to pacapavir despite the susceptible interference, and what can we do to mitigate this resistance evolution? And then, if there's a moment at the end, I'd like to just step back broadly and think about, what this teaches us more generally about these targeting these social traits in viruses. Okay. So I said that we built a model. Actually, my graduate student, Alex Robertson at the University of Washington, built a model And it basically conforms exactly to what I previously explained, except with equations on top of it. It's a discrete time. Initially, I'm going to show you a fully deterministic model, but we'll move to stochastic in a moment, in which you have some population of viruses that are genotypically, carry susceptible or resistant genotypes. And they get into cells according to the relationship between the density of the viruses and the number of cells that are present in the population. So there's some sort of Poisson entry process, wherein if you have more viruses, you get more viruses per cell. These viruses replicate with the opportunity for mutation. They produce capsid subunits at equal rates. We think that this Picapivir resistance doesn't seem to have a major cost of resistance. They assemble into these 60 subunit capsids, and then they package randomly the viruses or the genomes that are present in the cell for export. Then we think about the idea that the virus is able to be bound by Picapivir. And in order to try to understand how that works, we're thinking about this in terms of the probability of survival, or alternatively, the complement of the probability of neutralization as a function of the number of resistant subunits that are present in a given capsid. And so, on the one hand, you may have totally resistant capsid, and on the other hand, you may have some totally sensitive capsid or something in the middle. And we parameterize this probability of survival by looking at experimental data from cell culture experiments and found that it had a kind of shape like this. Now, some of you might be looking at the shape and thinking like, oh man, this actually looks a little bit strange, and that, the sort of promise of the susceptible interference is that, you should have to have a good chunk of of resistant viruses before you really start to see phenotypic advantages. But what we found when we looked at these data is actually a little bit more sobering, that actually even a small number of resistant particles did seem to confer some advantage right away. So we can see how that affects the results. So we wanted to use this model in order to see first, can we replicate experimental data? And it turns out that we can. So this is the data that we showed you earlier in terms of presence or absence of Picapivir, and simulations seem to do this just fine. It's not really surprising that this works out well, in part because we parameterize these curves based on these data, but it's nice internal sanity check. But we were really interested in doing is trying to use this model in order to ask about the process of resistance evolution. And so this is where we went next. So the first question, the first sort of promise of Picapivir is that we should be able to treat these viruses, knock down their population sizes without letting genotypic resistance increase in frequency, specifically because we think we're blunting the selection for genotypic resistance by disassociating the genotypes and the phenotypes. And what we found is that at high multiplicities of infection, our model predicts that the changes in resistance and frequency in just a single generation, this is genotypic resistance, were very small. Suggests that really resistance should not be evolving. And again, this makes perfect sense based on the sort of internal model of what's going on. Rare genotypic resistance should basically never feel its phenotype. However, what we learned is that, if you start looking at what this looks like across different multiplicities of infections, you get much more substantial changes in genotypic resistance frequency, even in a single round of replication. And the real reason is because what happens is that at these low multiplicities of infections, you get that most viruses are only infected by a single, most cells are only infected by a single virus. So if that's a susceptible virus, then all of these viruses are vulnerable to neutralization. Whereas if that's a resistant virus, then all of a sudden you're in trouble. Because what has effectively happened is that at these low multiplicities of infections, you reestablish this genotype-phenotype map, and viruses are able to feel their own phenotype, and selection is able to act much more effectively. So this is maybe not completely surprising, but what we were really interested in was asking what happens over time if you sort of allow a dynamical process that feeds back into itself. What is going to happen over the course of multiple rounds of infection if you're thinking about what should happen to this process intra-host? So we did these simulations in which we started populations at really high, ubiquitous sensitive viruses, and asked what is going to happen to the viral populations over time. And effectively, we took the output of one generation and then we inputted into a fresh cellular population in the next generation. And what happens is that in these initial steps, we get these huge drops in viral yield, which makes sense because Picapivir is working. And you can see that drug resistance basically hardly creeps up at all. However, once we have this big drop in viral yield, we get into this mode where viral co-infection is greatly depleted. Selection can see resistance, and then in subsequent generations, resistance can emerge much more readily. And what we think is happening is that this is offering a pathway to the virus to evolve resistance by first, going out of the conditions in which susceptible interference is able to mess up this process and into one where susceptible interference basically does nothing at all. And so this was one of the outcomes that we saw in the clinical trial, was widespread resistance evolution. But there was a second outcome that we were interested in trying to explain, which was this really early susceptible clearance. I should mention here that, if you look at this on a phase diagram, you see this really nice pattern where no matter what happens, if you start at high multiplicities of infection, the first thing that happens is the MOI goes down and then the resistance comes up. Okay, so in order to try to get at these susceptible early clearers, we basically did two things. We turned this from a deterministic model into a stochastic model in which all of these processes are happening, but we're sort of sampling from distributions and sort of instead of taking an expectation. And we added a model of poliovirus immune survival because we needed to be able to mimic the process of clearance by the immune system, which happens if someone has poliovirus. And what we found is that we could simulate clinical trial data that really matches phenomenologically these patterns that we saw in the clinical trial, which is you get early resistance or early clearance with susceptible virus or late clearance with resistant virus. And we can understand what's going on under the hood. So in these late clears, we get this pattern in which the viral yield drops, the resistant genotypes get associated with their phenotypes and rise in frequency before being cleared by the immune system ultimately. Or a situation among the early clears where we get these same stochastic bottlenecks, but in these particular lucky cases, you can get viral clearance when the population sizes are really small. So we think that the sort of single model that accounts for viral density over time does a good job of explaining potentially both of these clinical outcomes. And so I think that this, I'm missing part of the slide. I think that what we're kind of seeing is that there's this interesting feedback loop where viral social interactions are potentially creating these realized phenotypes like viral capsids that are more or less sensitive. And these realized phenotypes are feeding into the absolute fitness. And in the context of the absolute fitness of this population with a drug, it's sufficiently low that this is feeding into the population density in such a negative way that it depletes the viral social interactions in the next generation, which changes the realized phenotypes, where instead of having these mixture chimeric phenotypes, we all of a sudden see, fully resistant or fully susceptible ones. One of the results of this is that actually, I showed you that curve that looks like it's not very, doesn't have a lot of susceptible interference in it. It turns out that you can make a lot of modifications to that curve and still recapitulate this phenomenon, in part because what happens is that this decrease in density leads to the segregation of genotypes into their own cells, where the actual mechanism of this interference aren't so important. Okay, so with my last minute, what I want to tell you about is just, what we can do about this. So it seems like the big problem here is that we tank viral density. So we asked what could happen if you actually increase the degree to which viruses can survive pacapavir. So let's imagine that we could treat someone with less pacapavir or a less effective form of pacapavir. What should happen to the probability of viral survival? And we think that what this should do is basically permit more sensitive viruses to survive. And as a result, you might get some, continued interference in the longer run. And we found that this is actually exactly what happens. So here's what happens with pacapavir. It neutralizes the virus. But if you think about a 100 times weaker version of pacapavir, where we've just rescaled that fitness function, you can see that, your viral load drops much less precipitously. But as a result, you get this sustained competition between, it's not even competition, the sustained co-infection between resistant and susceptible types. And ultimately what this leads to, although I don't have time to show you in full detail, is that resistant viruses are really suppressed for much longer periods of time. And in fact, the viral population sizes as a whole over the course of infections are lower because you never get this full resurgence. So I'm going to skip through showing you those results and just sort of end with this idea that counterintuitively, reducing pacapavir's efficacy can lead to resistance and fewer replications. And I'm really excited about these feedback loops and thinking about the importance of viral co-infection when trying to design therapeutic strategies that can exploit this interaction between viruses. And I think that there's a lot to do here in a bunch of different arrows, like including thinking about more complex relationships, how we can think about viral social interactions going into realized phenotypes as a type of viral ploidy, and then ultimately, how we can manipulate population density in other ways that are maybe a little bit more complex in order to really exploit these social interactions. Okay, so I want to thank Alex, who's the grad student who did all of this work, and Ben Kerr, who's a collaborator on this project. And with that, I'm happy to take any questions.
Thank you. Any questions for anyone? Troy, is your hand up?
My hand is up and my hand is clapping.
Okay.
Thanks, Alison. That was super interesting. I wonder if you also started looking at things like multi-locus resistance. So in particular, when there's epistatic interactions between loci, you could imagine that during co-infections, even if one particular strain only has one of the relevant mutations and another one has the other, they can sort of compensate for one another through a co-infection, and what sort of consequences that might have for the spread of resistance that's determined by these kinds of epistatic interactions.
Absolutely. Great question. Yeah, I mean, I really like it in part because it sort of connects to this idea of like thinking about viral ploidy as maybe this is some sort of overdominance in which, you know, mixed infections of both of those different things can create phenotypes that are maybe only accessible in mixtures. So we haven't done this in the context of resistance evolution, but we have been thinking about this in the context of measles, where we think that there's actually some good evidence in measles brain infections that you know, mutations that both increase and decrease fusogenicity are actually working together in order to better permit the cell or viral cell-to-cell transmission that is necessary for measles to move through the brain. And I mean, I'm very curious about the extent to which, you know, you actually need two different types of these fusion proteins in order to potentiate those interactions, and or whether it is possible that you can get a single genotype that encodes the phenotype that you would want. So I think it's interesting to think about situations that should select for, you know, properly optimizing this thing where, you know, you could imagine you get two mutations on one background and, you know, you're good to go, you can spread by yourself, versus situations in which you actually can't even achieve that unless you have co-infection. And what are the situations that can select for that kind of scenario? And I think something where viruses are kept in close constraint, like in, you know, cells in the brain where they actually can't leave, is one situation where we might expect to see that kind of evolution.
Oh, that's very cool. It's like a sort of restricted movement selecting for cooperative interactions almost.
Yeah, what we think is potentially going on. There's more work to be done there.
Yeah, that's very cool. Thanks.
Sam?
Yeah, hi. That great talk, really interesting. So I want to sort of take this up a level. Co-infection in some ways implies that there's a population out there with differential infections. So what are the implications here for the dynamic at the population, the host population level, and uh, and you know what the right vaccination strategy might be or treatment strategy?
Yeah, I love that question too. That corresponds a little bit to some results I didn't quite get to, and actually some work that's still ongoing, which is that one of the things that we have seen is that these sort of lessened versions of bacapavir seem to knock down the viral load over time, but increase the amount of time to clearance because you're effectively giving up that opportunity for stochastic early clearance. So the question becomes, are you better off by waiting longer, or, you know, having these longer, lower viral load infections, or are you better off, you know, with these things that have this split outcome? You could either have really high viral load resistant infections or totally no infection at all. And I think that you know, trying to balance the you know, these different factors is really interesting in a public health context, and differs from exploiting these types of sort of moderate dose approaches in something like cancer, where you don't have to worry about onward transmission as a potential outcome. But I think maybe I'm just realizing now that maybe I slightly misunderstood part of your question. We're largely thinking about resistance that emerges de novo within participants here, though, because we assume that most of the viruses that are circulating are going to be susceptible, and the rate of mutation for these resistance mutations is like on the order of 10 to the negative fifth, like super, super high.
Thank you.
I also have a question. So this phenomenon that you're talking about, you know, makes sense for these viruses where there is, you know, quite a bit of cellular co-infection. Of course, you know, viruses also have, you know, things like superinfection exclusion and so forth. But I'm wondering more generally about kind of application of some of these ideas to actually, you know, pathogens, like non-viral pathogens, right? So for example, like in bacteria, I mean, they're not infecting, you know, co-infecting cells, you know, but I'm just wondering in terms of like bacterial mats, right? Could those actually have a phenotype, even if a single kind of phenotype, but which are, you know, kind of a combination of kind of the genotypes that kind of comprise these mats, you know? So have you thought about that, about, you know, applicability of these sort of, you know, phenotypic hiding ideas, to other pathogens that might not, be dominated by or might not have cellular co-infection?
Yeah, that's really cool. So I mean, we've thought about this a little bit in the context of just drawing analogs to strategies in treating like using antibiotics in pulsed ways that maybe maintain competition between sensitive and resistant types. But we haven't really thought about the idea of hiding within the context of bacteria. I know that there are definitely instances where, you need multiple co-locating types in order to produce some of, I'm blanking on the specific details, but I have kind of a clear remembrance of reading a paper, I guess not that clear a remembrance of reading a paper in which, mixed infections forming biofilms actually need both cellular components in order to produce a phenotype that I think gets maybe a little bit to Troy's question. But yeah, it's not something that we thought about at length in terms of like, but I imagine a lot of the same principles would
that kind of cycle diagram you had of the feedbacks, right? And I also something happens for other social traits as well, but I was kind of thinking that the timescales are super important there. And you're seeing both of these things kind of like play out on a concurrent timescale so that one influences the other. But that doesn't have to be true, right? You can imagine that the fitness consequences might be so fast that one timescale kind of like resolves itself before we even see it. So I kind of wondered if there might be whole classes of viral social traits that we never see because they drive themselves extinct ecologically.
Yeah, I think you're completely right. And, I think in Mahan's sort of nice example of calling out other work that is nice work that's going on in the audience. Yeah, I mean, I think that, some of the work that you've been doing on multipartite viruses and sort of how do you end up in these potential evolutionary dead ends that are harder to see by virtue of their harder to propagate is super exciting, sort of that viruses can do things that are locally good that ultimately select for worse fitnesses in the long term. Yeah.
Cool. Yeah, I wasn't necessarily thinking of my work, but yeah, thank you. I think yours
I
is
mean,
a
it's
better
definitely
example of that. But yeah, cheers.
the first thing that comes to mind for sure.
Jeremy.
That was a great talk. Sorry, just unmuting. I feel like there's some similarity though to some of these like earlier studies about just sort of the trade-offs of resistance evolution and sort of ecological competition with by the susceptible type, if you sort of tailored appropriately can sort of prevent the evolution of resistance. So in some sense, I'm wondering, like I can see that sort of the initially where that social effect is coming in, but whether there's sort of still like how much of an effect in the end it is having on this system if you just modeled it as just sort of a cost of resistance or something like that, and then assumed that they were competing ecologically in sort of a well-mixed population, you'd still get some kind of similar effect where you could potentially slow down the evolution of resistance by allowing the wild type to compete for longer with the resistant type.
Yeah, I think that's totally true. Thinking of some sort of relationship that is non-monotone, or I mean, relationship between, how strongly you select for resistance and just the degree to which, resistant things increase in frequency, and the stronger you make that selection, the more you're going to get that effect. Yeah, I mean, I think that we've begun thinking about the degree to which this differs from some of those scenarios. I think the major thing that we think about is largely that there's not direct competition. I mean, there is to some extent, but between the sensitive and the resistant types intracellularly, so much as a situation in which I think these things that are exploiting the growth of the sensitive populations, and especially the growth of sensitive populations that might have, a higher R naught in the absence of a drug, so that you could sort of allow these populations to slightly come back up and then, apply a drug again, knock both populations down, but knock the sensitive one down a lot faster, that you could be maintaining over longer periods of time, both of these for longer. I think that there are definite analogs to the work.
Cool.
I think that the major difference here is that we're thinking about a susceptible unit that sort of sensitizes the resistant ones as opposed to something that directly competes against them.
Right, right. Yeah, that's very cool.
Well, in the interest of time, let's move on to our third speaker, Chadi Saad-Roy.
Hi. Let me just share my screen. Hope it can work. Oh, so now do you see does it project well or no?
Yeah, we see it.
Okay, nice. Awesome. Great. Let me just hide these controls. Yeah, all right. Thanks a lot. Thanks for yeah, very tough acts to follow. Thank you, Katia, for the invitation. Thank you all for listening. So I thought hopefully I'll make it quick so we can get to the panel, but I'll give a brief overview of this new line of research that I've been thinking about on post-infection mortality. So I think this is, so just to think about this trait, we can think of pathogens that cause long-term harms to their hosts even long after recovery. And I think this is, though it's present in the literature for, you can look back and there's all these post-infection syndromes from a lot of different viral pathogens. But I think it's really brought to light with COVID, and long COVID is this kind of clear representation of this. And that also motivated many reviews. So there's many recent reviews studying these post-infection syndromes across many pathogens. So I think this is a clear example that pathogens can have long-term effects, especially after individuals have recovered. And so I guess the question is, what are the impacts of post-infection mortality on population dynamics, and what conditions may favor or hinder the evolution of such a trait? So to begin looking at the epidemiological impacts, we can consider a simple model where individuals are susceptible, they get infected, they recover, and then they can potentially get reinfected. And then in that class where they've been infected before, then they're potentially subject to slightly elevated rates of mortality, and call this post-infection mortality. And you'll notice here I also have, actually, let me make the cursor, the laser pointer, if I can. Anyways, yeah, there we go. So you can see I also have mortality during active infection as well. So to formalize this is what the model looks like. So individuals, SP, are those that are primarily susceptible. They get infected into I. Once they are infected, they recover at some rate, and then they're secondarily susceptible, SS, where they can get reinfected with some relative susceptibility epsilon. So if epsilon is 0, then you at an SIR type model, and if epsilon is 1, you're at an SIS, right? So you're as susceptible as you were before infection. In SS, you see this extra term, the mortality during SS, elevated mortality. And then alpha I is this mortality during active infection. So this is the crux of the model, and here's the reference for those that are interested in looking more. So I'll give you just a high-level overview of what happens. So in the long term, you can actually see that if you don't have post-infection mortality, the endemic equilibrium is stable, and that kind of makes sense. There's only one. So there's one endemic equilibrium, and it's stable. But if you have post-infection mortality, it can trigger sustained oscillations. So here's an example. So you have time on this x-axis and the fraction of initial population on the y-axis. I discarded the initial transient, and I'm showing you the limit cycle. And you can see here for an R naught of about 2.5, you have these cycles on the order of decades. On the right panel here, the right panel, I'm plotting this transition. So you can see what's going on. You have just the pair of the eigenvalues that have complex, that are complex. And you can see here you cross this threshold. And so the star is the one showing you the simulation. So you have this bifurcation happening. And as you increase this mortality after infection, you become stable again. And this is what you see here. So what's going on? Well, if you think about this, so first let's explain why is it stable if you have really, really high levels of post-infection mortality. Well, you have more individuals are dying immediately after recovery, so you wouldn't expect anything else. Now, why do you have these cycles? So if you think about the two pools of susceptibles, you have the primary susceptibles and the secondary susceptibles. Infection moves one from one to the other. But then the secondary susceptibles are subject to slightly higher mortality, which crashes the R effective, right? So your R effective goes below 1, the infection goes down, allowing the primary pool of susceptibles to build up. And then eventually the R effective crosses one again, and then so on and so forth. So this is the intuition behind what's going on here. And then just to show you that you actually can get pretty low, you can have very low rates of mortality after infection. So here's this parameter plotted here, time on the x-axis again, and primary susceptibles, secondary susceptibles, and I over the 3 panels. And here I plot essentially the long term for each transect, and you can see where the cycle appears, right? So fairly low rates of post-infection mortality. So now what happens if you have other traits? So I did stress the importance of potentially immunity after infection. So it turns out if you have a robust immune response, that's stabilizing. And that again makes sense. The secondary pool of susceptibles is contributing less to susceptibility and eventually just disappears. And that's what these 2 panels show. And if you have mortality during infection, that actually can accentuate the effect of post-infection mortality. So you need less mortality after infection with a little bit of mortality during infection to still get endogenous oscillations. So you can see here, there's with alpha I, mortality during infection, at 0, you have no oscillations. And this is with some mortality after infection in the background, right? And as you increase this parameter, your cycles appear. So this shows you that disease-induced mortality can synergistically complicate this dynamics. What happens with seasonality? So this is you can just make transmission seasonal function now, and you could get very complex dynamics. So here's one example. You actually see you get your endogenous PIM-driven oscillations back, but you also have this interaction with the seasonal cycle that makes things rather interesting and complicated. So yeah, we can explore this more, but yeah, we can do this afterwards or offline. So what about the evolution of post-infection mortality? So far we know that this trait seems to potentially cause complications for long-term prediction. What about the evolution of it? So we can write down a simple model to test this out and essentially assume that you have an endemic pathogen that doesn't cause post-infection mortality and ask when can it get invaded by one that does. So this is the cartoon, and next I'll show you and here's the reference. Next I'll show you the model framework. So this is basically what I showed you before: susceptible, primarily susceptible, infected, secondarily susceptible. This is in the absence of any post-infection mortality. And now you can ask, when can a mutant that causes some post-infection mortality invade this endemic pathogen? And so now you have star, it's the mutant, not star is the resident. And then you can do a little bit of analysis and show that unlike classic virulence, so mortality during infection, PIM, post-infection mortality, is neutral and not selected against in the absence of other trade-offs. So that's kind of interesting because it doesn't come into the basic reproduction number, right? But if the relative susceptibility to reinfection is independent of post-infection mortality, so now it doesn't matter, this epsilon doesn't depend on PIM, then all it matters is how post-infection mortality affects other character traits, other pathogen traits, such as the recovery, disease-induced mortality during infection, or transmission. And now the question is, what happens when post-infection mortality affects the relative susceptibility to reinfection? So then you can actually obtain a threshold for this quantity, essentially this susceptibility PIM relationship that'll enable PIM to evolve. And you can show that if you have an increase in R naught is driven, an increase in PIM increases R naught, then the threshold's always below 0. And the magnitude of the threshold will depend on the baseline values of other parameters. So here I plot the thresholds, this epsilon prime, right? So this relationship between how epsilon changes at 0 when you have 0 post-infection mortality as a function of post-infection mortality. So here's that's the y-axis. The x-axis is this transmission rate with no post-infection mortality. The columns are baseline values of mortality during infection. And then this row is this change, basically the relationship between transmission and post-infection mortality. So we're assuming that the R naught's increasing, which is basically this example here. And you can see that for different values, baseline values of epsilon naught, so this relative susceptibility to reinfection, the threshold can be different, right? And the ordering of the threshold changes with how much baseline transmission there is. but it's always below zero, and that we know analytically. So it's showing you that these actually, depending upon the transmission rates, there's very little, it's very easy for post-infection mortality to evolve. Then you could do something that's maybe a little bit more interesting. Contrast during infection mortality with post-infection mortality. So you can do the equivalent calculations for invasion with during infection mortality. And there you can compare and contrast them side by side. So here we've got the columns now are this baseline susceptibility to reinfection. The rows are again this change in transmission. And then in each type, you have the mortality type. So during infection mortality or post-infection mortality in the colors. The y-axis is the threshold, and the x-axis is again the transmission rate with no mortality. And you can see that as expected, sometimes during infection mortality, you can still even the threshold can be positive and you can select against it, whereas post-infection mortality is never selected against for thresholds that are positive. Another interesting note is that the relationship seems to, with PIM, it's always this increasing function of transmission, whereas with DIM, here it starts off as a decreasing function, and then you have this slight change, and eventually for high enough baseline relative susceptibility to reinfection, starts increasing again. So there are these interesting differences, but again, note that it's much easier to evolve post-infection mortality than during infection mortality. Oops, now the cursor is not working. Yeah, and you have some qualitative differences, but at the baseline, that's the end result here. So the final question that I'd like to think about is how can we characterize post-infection mortality? So this has all been highly theoretical so far, but we can think about different studies that might help this. And so actually in a cohort study that we conceived and we laid forth in this perspective in 2022, I think a lot of the a lot of the data that one could get from such a cohort study could be very relevant for post-infection mortality. So here our idea is that individuals will be tracked as they move from susceptibility to infection to recovery to waning immunity and to reinfection. Their sequences, viral loads, etc., would be considered. And then at each step for an infection or reinfection, the household transmission data would be collected to estimate how transmission changes with immune status. And then obviously there's this question of age structure as well that we could investigate with such a cohort study. And now I think if we have such a cohort study, we can start answering some important questions about post-infection mortality. What are the rates? How does it correlate to other pathogen traits? So this would be really important, I think, to be able to characterize this trait in a less theoretical fashion. So to conclude, hopefully I've convinced you that post-infection mortality can cause sustained epidemiological oscillations in the simplest possible model. Unlike during infection mortality, post-infection mortality is neutral in the absence of other trade-offs, and it can readily evolve. So some remaining questions. So once emerged, what are the longer-term evolutionary outcomes of post-infection mortality? How do repeat infections affect the evolution of post-infection mortality? And I've been in parallel work really interested in individual behavior and decision-making and how that affects epidemiological dynamics. And so does post-infection mortality affect individual risk perception of an infection? And if so, how does it shape human behavior? And so thank you to the co-authors, the funding, and everyone else for listening. And as with Mahan, I will put a shameless plug. So yeah, if anyone's interested, please contact me. And then now I can go back to the acknowledgements. Thank you very much, and hope that was somewhat interesting.
Great, thank you. Any questions, Joanna?
So, do you have any predictions across viruses? So I'm thinking, that a virus like measles clearly has, very high levels of post-infection mortality. That we do, I mean, it's imperfectly known for a lot of things, but it's not that we have zero data, comparative data. Is that do you have predictions if we could make that data good enough?
Yeah, that's a great question. So I think epidemiologically, because like if you have an acute immunizing infection that doesn't lead to susceptibility again, then the dynamics won't be affected by that. I think from an evolutionary standpoint, it would be pretty interesting to look at. But I guess that, yeah, so to answer your question, it would be really interesting to look at. I haven't done this myself yet.
Question. So you didn't mention immunopathology specifically, but a lot of it seems like post-infection mortality is a consequence of kind of immunopathology, oftentimes, not always, but oftentimes. And I'm wondering in terms of kind of your thoughts about, okay, well, if there's not kind of direct selection, kind of in the case of kind of it's kind of neutral post-infection mortality, in the context of immunopathology, what do you think it is about kind of let's say higher level of viral genotypes that kind of yield higher levels of kind of immunopathology, let's say, what is it about there kind of within host dynamics that might confer higher kind of, R naught values?
Yeah, that's a great question. So I don't have an answer, but I think like doing, yeah, obviously this is the 0th order first cut. I think we need to do some within host models or cross-scaled models to really try to answer these questions. I think that would be really important. Yeah. And we can talk more about it.
Any other questions? Okay, so maybe we can start kind of a panel discussion. Joanna, before we start kind of this relatively short panel discussion, can you, do you want to make an announcement potentially about the Chicago meeting?
Yes. So there is an in-person meeting in Chicago, June 2nd to 6th, but we are going to try and make it as accessible as possible also for remote participants. The mornings in Chicago time will be plenary talks that will be streamed, and the afternoons will be breakout discussions of various kind, and some of those will have the option for remote participation as well. So watch out for your inbox for invitations. Jen will be sending something out. It's not live yet, but we will be setting up a system, unless Jen wants to cut in and say that it's already set up. But we will soon be setting up a system where you can sign up for remote participation. If you suddenly find, if you're not signed up to come in person and you suddenly find you can be in Chicago after all, Jen is also a good point of contact. We've got cancellations, there are still in-person possibilities to participate. Not so much to present except for a poster, but Jen can still add you to the participants. Jen, did I leave anything out?
No, and it is not set up yet, so hopefully more coming about that by the end of the week or by next week.
Yes,
but I would also, oh, sorry, go ahead.
No, go ahead.
Okay. If it's quick announcements time, I'll just give a quick announcement here for next week, next Monday. We have another interview, Sally Otto and James O'Dwyer interviewing Robert Holt. So it's premiering at 10 a.m. Pacific time if you want to see it live. Otherwise, it'll be on the YouTube for eternity. So hopefully see you there.
Thank you. Okay, so for this panel discussion, I mean, everyone can be involved here. So maybe I can just start off with a question, but if we make this more of a conversation between the people who gave talks as well as anyone who's here, that'd be great. And again, kind of happy for anyone to kind of ask a broader question. So I have a question about so all of you talked about kind of viral adaptation, also kind of barriers to adaptation, and I'm wondering in the context of treatment both at the individual level as well as control strategies at the population level, which include kind of pharmaceutical interventions as well as non-pharmaceutical interventions. I'm wondering about kind of what all of your thoughts are about how far away and are we from really thinking about evolution in terms of in terms of the design of those treatment strategies and control strategies, and even whether in terms of not even being aware of things like viral resistance evolution and things like that, but even in terms of thinking about when treatment control strategies may no longer work because of viral phenotypes that evolve, how close are we towards using theory to design kind of evolution-proof sort of treatments and control strategies? Is that yeah? How far do you think we are away from that? Don't all speak at once.
More of Alison's related. I think Alison, if you want to go first.
I mean, I don't know. I feel like to some extent evolution proofing feels like a little bit of a fool's errand. I think it's impossible to do it completely. I mean, I think the best way to evolution proof is eradication. I mean, in some ways, like, finding effective strategies to ensure that you entirely, if you bring a population size to zero, you have no new genetic novelty, and that seems to be the only way to really completely stop the mutation rate. But I am excited about this idea of using, clever applications of strategies. I guess I think more about pharmaceutical interventions that sort of get us closer to that, either in terms of, exploiting shared phenotypes or treatment strategies that kind of play populations off themselves, like bacterial cases that we were talking about, or viral cases. Or also things that just, are strong enough interventions that they limit the production of mutations to the greatest extent that that's possible. But it seems like, at least in my understanding, that the only way to stop evolution is to get rid of a population altogether.
Can I ask Alison, can I ask you a question, as Troy here, about your results in sort of this question that Katia asked? What about the idea of dosing with susceptible nuclear capsid?
Yeah, I mean, I think this is something that people are thinking about. The idea that, you could be applying additional sensitizing, like, viruses themselves as a sensitizing agent. I think that a place where this is maybe being most seriously explored is in this concept of therapeutic interfering particles, in which you get, defective viruses that you are adding to a population in order to try to, like, you are hoping that those viruses are going to persist, and ultimately, I guess our definitions here would be that something that is replication incompetent on its own, but has a high enough R naught that it's going to spread within the context of a population, that if it is outcompeting other viruses that are present, that it could ultimately lead to a situation in which it drives the replication competent viruses extinct. But then also, once that has happened, those viruses are not able to themselves continue to replicate because of, missing some crucial protein or component. I think even in those cases, though, we need to be really careful about, the possibility that if you don't completely eradicate a population, then you might be getting yourself into the same sort of bind that we explored in our study, where, if your viral population density drops, then you run into challenges with, just resurgence of replication competent viruses again. But yeah, I do think viruses as their own treatment is a potential idea. I think the worry there is just, are you accidentally giving the virus more substrate for evolutionary novelty by, making population sizes bigger in a way that they can, become virulent again? Asher might have additional thoughts on therapeutic interfering particles if he wants to chime in here.
Well, one of the interesting things for TIPs is that people have made the claim that they are evolution proof. And I think it suffers from all of those things that Alison mentioned. But I wonder if maybe one way of reframing evolution proof could be slightly less evolution prone, right? Like, it doesn't have to be bulletproof. It just has to be good enough for our purposes.
Maybe switching gears a little bit towards this topic of vaccination, and maybe also I was just thinking in my head, a lot of things came through my head, like in terms of understanding within host versus between host evolution and the interplay between the two. I think there's a lot of scope for doing interesting work, which a lot of people in the audience are doing that work. But yeah, understanding these different evolutionary processes and how they manifest in the population generally. But like in the context of vaccination, for instance, thinking SARS-CoV-2 here, I mean, you look at the spike protein, you look at the receptor binding domain, the N-terminal domain, those are the regions where we see clearly there's evidence of accelerated evolution. And surely that has something at least to do with treatments putting a strong selective pressure in that region. But vaccines, I mean, you have to be careful here. Vaccines are, you know, don't target specific regions. They're more broader in terms of the regions of the protein that they target. Nevertheless, I think there's this talk about pan-corona vaccines, for instance. So you would try and isolate regions of the protein that are conserved across multiple pathogens. But then if we want to now switch, say, from an S1 subunit vaccine of SARS-CoV-2 to now targeting spike subunit 2, S2, which is more conserved at the minute, it would then also lead to a diversifying adaptive evolution in S2 subunits, which kind of goes back to what Alison was saying. I think it's a bit tricky in terms of, also this shifting of the landscape from, fitness advantage of, certain regions to now being, as a result of treatment, being pushed towards another region. And yeah, understanding that, I guess, is also an important, relevant aspect of all of this.
So I guess building on Mahan's point, I guess one can also think about all the work in the big literature looking at influenza universal vaccines. And I think that might be also this idea of evolution proof or reducing the likelihood of future variants. There might be interesting insights there. Although I guess the question always was, what happens when you target other places? Yeah, that aren't as immunogenic. Yeah. And is there a way to avoid that?
I guess an interesting kind of theory angle on that kind of question is thinking about the influenza question. You can do kind of deep mutational scanning approaches in the lab, right? And you can, in principle, predict where it's going to go and think of some kind of cocktail, that kind of thing. But you presumably, at least right now, can't do that for everything you would want to do it for, right? Like it's expensive, it's hard, not many labs can do it. So possibly a role for theory is kind of honing down that parameter space to have a better idea what we should focus on. I don't have any ideas there. Just
so maybe I can move on to another question. So in terms of theoretical understanding, there's work of course kind of last few years and also before that trying to kind of get an idea of kind of what types of evolutionary or what types of phenotypes we should see evolve, right? And of course kind of starting with kind of work way back when about, well, we shouldn't expect evolution towards avirulent infections, right? So I mean that's kind of the very, very early work, right? And then of course all the evolution of virulence work, and then should we expect kind of an increase in the transmission rate, or should we expect more antigenic evolution, or should we expect evolution of becoming potentially less symptomatic, or evolution of a pre-symptomatic stage, right? So we have some idea of what phenotypes we might see evolve in a certain for a certain viral system, right? Depending also on kind of its life history. I'm wondering about kind of theory to actually anticipate the time scales of kind of some of those phenotypic changes and adaptations. Is that obviously kind of we know the mutation rate kind of many viruses are kind of at the mutation rate at kind of in terms of every replication cycle, within just every time there's a replication, but how difficult is it going to actually be able to predict or anticipate kind of the time scale of adaptation, and how much do we actually need to kind of know about underlying kind of infectious disease population dynamics to actually be able to really have a good understanding and good kind of predictive power of the timescales of adaptation.
I think Chadi might have a lot to say about this, but maybe I'll just quickly say a few words about this topic, because I keep referring back to SARS-CoV-2 early 2020, where people were saying, oh, it's just gonna eventually plateau and, in terms of exploring its fitness landscape. And I'm like, really? Are we sure about that? I mean, it looks to me like there's a lot of options for the virus to acquire mutations. And how much can this be pushed in terms of do we expect to continue seeing these saltations or not? And a lot of people would then say, I mean, literally people just waiting, saying, "Oh, it's been 9 months, we haven't seen anything yet, so I guess it's been saturated now. We're good to go, guys." And then boom, another one, another one. So I think a lot of it has to do with, yeah, just being blindsided by understanding uh, pretty much anything about long-term evolution of these viruses. I think a lot of it has to do with not understanding host constraints, which I think you alluded to. The immune system, how does that put a pressure on how the virus explores that fitness landscape? Um, and how would that manifest over long time scales? Because here's the point. I don't know if how many of you have heard of this term called endogenous viral elements, like the fossil records of retroviruses could like integrate into our genomes and stuff. So you look at an lentivirus, like a family of HIV, which evolves like crazy, right? You look at an integration event, indigenization event from like 1 million years ago. Look at the virus, and it's like only 10% or 20% more divergent with respect to the modern day HIV. And you go, there's no way, right? Given how fast HIV evolves, that you would see a fossil record that is only 10% divergent. Obviously, it has to do with these host constraints that sort of goes back to my point about time-dependent rate effect. I don't want to ramble. I guess I'm just trying to say this host constraint work in very peculiar ways over longer time scales that are not trivial to us in terms of understanding them.
Yeah, and I guess building on Mahan's point, like I think what's pathogen, what's driven by the pathogen versus what's driven by the host is also important, I think, in this question.
Mhm.
And yeah, I think often we might think one or the other, but it might not be so clear. But I guess Mahan diverted to me, but I would love to hear like what other people like Troy or someone else who's thought a lot about this for many years would have to say.
Well, I don't think I have any answers, but I guess to me, uh, uh, the things that evolved, the high transmissibility that evolved during COVID, and what happened initially in terms of that, I don't think was that surprising. But what I thought was really surprising was how fast it happened and the way it happened genetically. I mean, the huge suites of weird things that were combined together to make these new variants. I think that was, I mean, to me, that was, I would never have guessed that that would be the way things work, right? I mean, I sort of have in my mind the kind of Fisher's model kind of idea of adaptation. And so there's big changes early on, but I mean, eventually you get closer to something where the smaller changes are the ones that invade. But um, yeah, I mean, I think the selection on transmissibility is clear. Virulence, impossible to say, I'd say. And the underlying genetics, I don't know. And the time scale catch it, all that seems really hard.
What about some of the like easier, better studied cases? I mean, I think probably like flu antigenic evolution is maybe the best studied and one where you kind of you see multiple genotypes circulating at once. And you kind of even can fit a curve to how fast one is taking over the other. So to me, that seems kind of like the simplest case. Do we even understand there, like if we increase vaccination rates by 10%, how that will change the rate at which antigenic drift is happening? I'm not sure. Maybe we do. I don't know.
I don't think we understand them mostly because, even with vaccination, I mean, then there's, it's about, the level of immune pressure, right? That's going to impact, the selection coefficient, right? And I think that's still unclear because of all the imprinting effects and so forth. I mean, that's a really difficult problem. And then of course, like, I mean, there's one aspect of it is like if you have, two variants in the population level and based off of kind of, data, can you anticipate kind of going forward based off of past trends, and that's kind of like weather reporting, so that things can come in this direction, east to west, at this rate, and then it's going to be rainy here on Tuesday, right? But I think it's this question of like, okay, well, without actually having those pre-existing lineages already kind of and having some data on them, can we, and maybe it's getting at kind of, okay, do we, with antigenic clusters, at least until, 2004, something like that, we knew for H3N2 that these antigenic clusters occurred every 2 to 8 years, right? Would we be able to, why is it 2 to 8 years, right? Should be faster, should be slower, would we be able to, and of course, it seems like antigenic evolution for SARS-CoV-2 is very different, than it is for flu, but would we, what is those time scales of kind of these antigenic changes? Can we predict that? How do we understand that? And I think there's still a lot of theoretical developments going on in terms of like trying to not understand which antigenic variant will arise or which one will outcompete the other, but just kind of, these sort of statistical properties of the system, right? How frequently or how, do we expect antigenic turnover to occur? And then, of course, like, is there diversification, overall antigenic diversification, or is it just kind of limited antigenic diversity at any point in time?
If you ran out of time, can I say a few words, or are we supposed to just say goodbye now? I don't know.
You can end it, Mahan.
Yeah, no. So then I'll shut up. Don't worry about it. We can talk afterwards. It's fine.
But I will say that this idea that like combining this idea of high resolution experiments where you can sample local fitness landscapes combined with these sort of forward predictions given conditional on these variants being present, what do we think is going to win? I think it's really promising, although it doesn't help us with Torres' problem of like what happens when you get 20 new mutations and you don't have the representative local fitness landscape.
All right. With that, let's thank the 3 speakers again, and thank you all for joining, and hopefully see you on Monday.
Thanks so much for organizing, Katia.
Yeah,
Thank you very much.
Thank you.